Thank you. I hope that you're not too bored of listening to me after presenting in the previous panel. So let me explain to you what TiGenix is about, hopefully. So we're a public company, so I'm going to be making, knowing myself, plenty of forward-looking statements, so you should not take them into consideration and read the whole documentation that appears in our webpage. So what is TiGenix? So we are a small biotech company based in, actually headquartered in Leuven, but with most of our operations in Madrid. About 70 employees, pretty liquid, very small market cap of about 170 million, depending which day you look at it. Pretty liquid, with 6 analysts covering the company, and actually with one very, very large shareholder, Grifols, that holds 21% of the company, then Novartis and Genetics, which is a company from which we acquire a platform, have about 3–4% each. And we had 23 million euro left in June. We burn a little bit less than 2 million euro a month. So you can do the calculations. We have enough money just to go until the mid of next year. And that may be the reason why we're so interested in coming here. What do we have, and why do we believe that TiGenix makes an interesting investment opportunity? First of all, because we have announced in the month of August the first ever positive phase 3 trial with a stem cell asset. CX601 has demonstrated statistical superiority to placebo on achieving combined remission in complex perianal fistula in Crohn's disease patients. And I will show you in a minute. This is a very large indication. We have a product that is very well protected, and we're expecting to file for approval in Europe in the spring of next year, aiming at getting approval in the second half of 2017 and launching ourselves in Western Europe thereafter. In the US, we didn't have the money to do a second phase 3 in parallel, but we've been advancing as much as possible with the FDA. So today, as well in August, we announced that we had a positive SPA, so we know exactly the phase 3 that we need to do in the US to eventually, together with a European phase 3, file for approval. We're in the middle of doing our tech transfer to Lonza, which will manufacture for us for this phase 3 clinical trial. Again, we have full commercial rights of CX601 in the US, and we're planning on conducting this confirmatory phase 3 ourselves. So only having a product that is ready to file with significant data and ready to go into a confirmatory phase 3 trial in the US should be sufficient to convince investors that we are a strong animal. As you know, I challenge everyone to find companies worth 170 million with that sort of a product in their pipelines. But on top of that, we have 2 very interesting assets that I think provide significant upside. One of them I'm not going to talk about today is our intravenous administration of these allogeneic adipose-derived stem cells for the treatment of severe sepsis. We're running, as I mentioned, we're going for large trials, so we're running a double-blind placebo-controlled trial in 180 patients in severe sepsis across Europe. And we announced last week that the Horizon 2020 program has just granted us 5.4 million euros to cover part of the cost of going with the trial. But the one that I will cover today is actually AlloCSC01, which is our leading asset out of our cardiac stem cell platform, again allogeneic, which we're developing for preventing congestive heart failure, treating patients immediately after a myocardial infarction. We are in the middle of a phase 2 clinical trial, again randomized double-blind trial. We have announced phase 1 data that I will show in a few minutes, and we will have preliminary 6 months data of that product in the summer of 2016. So major things happening in the next 12 months. So let me go to the 601. 601 is a product that we manufacture out of a liposuction of a healthy volunteer. You go to a liposuction clinic, you explain that you pay for the liposuction to those that give you the fat, and you have fat of 110% of the people there, because the nurse volunteers as well to get a liposuction done. Out of that liposuction, we just select the right adipose-derived stem cells, we grow them, we cryopreserve them, and we have them ready to be shipped when needed. In terms of scalability, one liposuction allows us to produce enough cells to treat 2,400 patients. So you can understand the scalability and the reduction in cost of goods versus an autologous product. What is a complex perianal fistula? Unfortunately, not very glamorous disease. I will not put pictures nor videos, but just this small graph that shows one of those fistula, this connection between the rectum and the skin through the non-anatomical channel. Very common complication of Crohn's disease. As many as 10% of the Crohn patients will suffer from these fistulas, and when they compromise the sphincter, when they are recurrent, when they have abscesses, they are considered to be complex. Today, there's no real therapy that will close those fistulas in those Crohn's disease patients that suffer tremendously from the disease. So what happens today when a patient presents with this complex perianal fistula in the doctor? They go through a pretty severe cascade of getting antibiotics to get rid of the infection, then immunosuppressants, and then very quickly the majority of the patients get infliximab, Remicade. Why? It's the only anti-TNF that has the indication of fistulizing Crohn's disease, despite very low efficacy in terms of closing the fistulas. Now, it's a very potent anti-inflammatory agent, so the patient feels better, some of the fistulas do not drain anymore, and despite the very severe side effects associated with the long-term use, you do market research with the opinion leaders, they tell you they need to offer something to the patients, so they keep treating them with a long-term use of infliximab. Some patients may go to Humira, and in some very desperate cases, you will end up with surgery. Again, in Crohn's disease patients with your tissue in bad shape because of the chronic inflammation, surgery is the last resort. About 1/3 of the patients that go through surgery will end up with anal incontinence. There's a very recent publication that reported that patients with complex perianal fistula that go through surgery have, on average, 7 surgical interventions. That gives an idea of the need of those patients. So what do we offer? We offer a single-dose product. All you need to do is, on the normal treatment of the fistula, which is, strong cleaning of the internal tract, and then identify the internal opening, stitching it, and then you just put half the dose of 601 around the internal opening with a normal syringe injection, half the dose in 4 or 5 points across the fistula tract. Once you send the patient home, they don't come back. Infliximab, you go back every 6 weeks for a 1-hour infusion. So what's the data? We did a very large randomized double-blind placebo-controlled trial, 289 patients, 8 countries, Western European countries and Israel, 50 centers. This was designed in agreement with the EMA to ensure that if positive, we could file with this single phase 3 clinical trial. Very important, we were forced to choose the toughest endpoint possible, which is combined remission, looking at the fistula being closed, despite pressing around to make sure that it was not suppurating, plus ensuring through MRI that there's no collections, no abscesses inside the fistula tract, and that therefore your fistula is properly closed. And at 6 months, so we gave the injection, the endpoint is measured 6 months later. And again, pretty important is that all the tracts were treated, so we allow patients, if you go to the following slide, we allow patients, adult patients, men and women that may have up to 3 fistulas, and we treated all of them in one go with 601 or placebo, double sham design. The fistulas had to be draining, the fistulas had to be refractory to standard of care, and we kept the standard of care, so this was an add-on design. We wanted to see what was the effect on top of whatever doctors are using today. Very important, very good balance between the study groups. The only thing that was unbalanced, hopefully it works, yes, is that we had a significant higher proportion of multiple fistulas in the 601 group. This imbalance, which was against 601, was good, because when I show you the data, if it was the other way around, we will be giving a lot of explanation. So it was good that it worked against us. And with that, we announced the 24th of August. By the way, after 10 years of working, we announced the positive data the day China collapsed, and the market as well. We announced these very positive results in which 601 achieved statistical superiority over standard of care with a p-value below 0.025, which was the hurdle to pass. So extremely good results and very significant results for the patients. Not only that, but if you compare, and remember, patients are being treated with other drugs, so there's other side effects associated, there's no difference between adding placebo or 601 to the underlying treatment. So we're adding a huge amount of efficacy without adding any side effects to the allogeneic use of these adipose-derived stem cells. So with that, as you can imagine, we've been 5 times in front of the EMA. I said go and go often. We've been twice for preclinical. We've been twice for CMC. We went one to ensure that the phase 3 clinical trial will be exactly what will be required. We're having our pre-submission meetings as we speak, and we're aiming at filing in the first quarter of next year, getting approval in the 2017. Again, we will partner Southeast Asia, we will partner Japan, we will partner Eastern Europe. We want to keep Western European countries, at least for the time being, for ourselves. In the US, as I mentioned, we went to the FDA, and what we have achieved, I think, is remarkable. Having an SPA, a positive SPA on this indication was considered by the opinion leaders in the US a major achievement, and that allows us to know exactly the clinical trial that we need to do in the US to get approval. And impressive enough, is exactly the same primary endpoint, but with a lower p-value, as this will be the second of the 2 clinical trials. So it's really a confirmatory clinical trial. Given the low clinical risk associated with it, we've decided as well that we will raise the money to do the clinical trials ourselves. So that's 601, and that should explain, I believe, a significant increase in the value of the company. Again, find me another company with such an asset ready to be filed, ready to go into confirmatory phase 3 in the US with 170 million market cap. But not only that, we have this asset, which you're going to see is pretty remarkable. What we do here, people used to die out of myocardial infarction. Today, people get a stent, and very few people actually die. What happens is that you get a scar, and that scar produces damage and produces the heart to try to compensate by increasing its size, ending up with congestive heart failure, which is today a major issue in the developed world. What most of our colleagues in cell therapy are doing is trying to go here, injecting the cells in the myocardial part and trying to get the heart to regain function. We have a completely different approach, which is to inject the cells here and try to prevent the damage to happen. And I'm going to see you why we're so convinced that this is the right approach, at least with ourselves. What we've done is animal experiments in pigs. For those of you that are not familiar, pigs are the best model for hearts. And what we've demonstrated that in a dose-dependent manner, we can reduce the cardiac remodeling of the heart, statistically significant. We can increase the ejection fraction of the heart of the pigs, again, statistically significant. And we can, this is histological cuts, that we can reduce significant, the blue is the scar. We can significantly reduce the scar of the heart after receiving this single dose. So that's great because it happens in pigs, but you want to know what happens in humans. So do we. So we did, again, with an H2020 grant, this phase 1-2 clinical trial in patients with a pretty large infarct size. 5 days after the infarct, we give them 1 dose of these allogeneic cardiac stem cells, and we're going to be looking at mortality, of course, and safety, but we're going to be looking into what happens in the heart and what happens on the functionality of the heart. Again, the data of the trial, 6 months data on all the patients, randomized double-blind, will be presented, will be available in the summer of next year. So a huge value inflection point for the company at that point in time. And the data we have today is the data on the 5 patients on the phase 1 part. This is non-controlled trial, so please bear with me, only 5 patients, no control. But it was presented in a poster in the European Society of Cardiology in August this year. And what we've reached with just 5 patients, making the comment of Sami before, it depends on the difference to reach a statistical significance, is that with only 5 patients, we have a statistical significant improvement in the infarct size, not only in volume, but relative to the left ventricular, and that you would expect an improvement also with just doing nothing. What you would not expect is a significant improvement in the ejection fraction. And that 15% relative improvement in the ejection fraction is unbelievably high. So again, It's only 5 patients, no control, very few conclusions that we can draw. We need to wait for the 6 months data on the control part, but we believe that there's significant upside here. So in the last 10 seconds I have, this is just to show everything that is coming, the end of the recruitment of the clinical trial in cardiac and the start of the sepsis trial that I was mentioning, but the most important one will be the filing in the EMA, hopefully to be the first allogeneic product to be launched in Europe in 2017. Thank you.